On His Majesty's service. Photoreceptors. Photoreception is a mechanism of light detection that leads to vision and depends on specialized light-sensitive cells called photoreceptors, which are located in the retina of the eye. There are two types of photoreceptors, namely rod cells and cone cells. The retina contains approximately 120 million rod cells and 6 to 7 million cone cells. They consist of three parts, rod or cone respectively, cell body, and axonal process. Each rod or cone, in its turn, also consists of three parts, the outer segment, the connecting stalk, and the inner segment. The outer segments are the sites of photosensitivity. In rods, the outer segments are long and cylindrical. In cones, they are shorter and conical. Both of them possess up to 1,000 tightly packed membranous discs that lie adjacent to the pigment epithelium of the retina. These discs are formed by plasma membrane invaginations and move proximally to distally toward the tip of the outer segment, where expanded discs are shed and phagocytosed by pigment cells. The formed rod discs lose their continuity with the plasma membrane. In cones, the connection of discs and plasma membrane exists throughout the entire length of the cone. The inner segments of both types of light-sensitive cells possess organelles associated with active protein synthesis, such as rough endoplasmic reticulum, free ribosomes, Golgi complex, and mitochondria. The connecting stalk between the outer and the inner segments contains modified cilium. The components of discs are synthesized in the inner segment and transported along microtubules of cilia toward the inner segment. Cell bodies of photoreceptor cells form the outer nuclear layer of the retina. Their synaptic processes form contacts with processes of bipolar and horizontal neurons. Description of the retinal structure and physiology is beyond the scope of this presentation. This material is presented in the section Sensory Organs of histology textbooks. Here I will focus only on the structure and function of photoreceptors. Rod cells are responsible for scotopic vision, that is, twilight monochromatic vision under dim light conditions, and cone cells for photopic vision, that is, detailed color vision under daylight conditions. Rods are more sensitive than cones and can generate a response from even a single photon. Membranes of photoreceptor cell discs contain photopigments. In rods, it is rhodopsin. In Greek, rhodon means rose and opsin means sight. Rhodopsin most strongly absorbs green-blue light and seems reddish-purple. That's why it is also called visual purple. Rhodopsin is G-protein coupled receptor that consists of protein molecule scotopsin, one of the large family of opsins, and covalently bound cofactor retinal aldehyde of vitamin A. Photopigment of cones, initially thoroughly studied in the chicken retina, was named iodopsin but in humans, photopsin is a more appropriate and frequently used term. It also contains retinal and one of three opsins: long wavelength sensitive opsin, which has peak sensitivity in the yellow-green part of the electromagnetic spectrum, but usually is called red opsin because it is more sensitive to red than the other two human opsins. Middle wavelength sensitive opsin, having peak sensitivity in the green part of the electromagnetic spectrum, and short wavelength sensitive opsin, having peak sensitivity in the blue part. According to the type of photopsin, cones are classified as L cones or red cones, M cones or green cones, and S cones or blue cones. Although each cone type is most sensitive to wavelengths of light at the peak sensitivity, all photoreceptors also perceive other colors owing to the overlap with colors perceived by other cones. The distinction between visible colors depends on the ratio of the three types of stimulated cones. Light-sensitive cells are not evenly distributed across the retina. Cones are present at a low density throughout the retina with a sharp peak in the fovea. Rods are present at a high density throughout most of the retina with a sharp decline in the fovea. In the central part of the fovea, the foveola, there are no rods at all. Most of what is known about the process of phototransduction has been obtained from studying rod cells, although the phototransduction in the cones in general is similar to that in the rods. In the dark, the level of cyclic GMP in the rod cell is high. Many cyclic GMP molecules are bound to the cytoplasmic surface of cGMP-gated cation channels, causing them to be open. This permits sodium and calcium ions to flow into the cell, which is therefore in a depolarized state with a membrane potential of about -40 millivolts, and the rate of neurotransmitter glutamate release in the synapse is high. The absorption of light by rhodopsin initiates the phototransduction cascade, that is, the conversion of light energy into a nerve impulse that is transmitted to the visual cortex in the brain. When the rhodopsin molecule absorbs a photon, its configuration changes from 11-cis retinal to all-trans retinal, which triggers changes in the opsin, leading to the formation of metarhodopsin II. The latter stimulates intracellular messenger G protein called transducin, resulting in the liberation of its alpha subunit, which activates cGMP phosphodiesterase. It converts cyclic GMP into GMP, thus reducing the number of cyclic GMP molecules that can bind to the cGMP-gated channels, which in turn leads to their closure. The potential across the rod cell membrane becomes more negative, leading to decreased glutamate secretion that is detected by bipolar neurons. Biochemical cascade initiated by one photon capture provides quick signal amplification. It was shown that activation of one rhodopsin molecule ultimately leads to the closure of about 200 cation channels which causes a decrease in the membrane potential of 1 mV. Progressive increase in the intensity of illumination leads to hyperpolarization of the rod cell, with a maximum membrane potential of -65 mV when all cation channels are closed. Thus, photoreceptors do not exhibit action potentials, since light activation causes a graded change in membrane potential and a corresponding change in the rate of transmitter release onto postsynaptic neurons. Rapid recovery of the photoreceptor after light activation is important because it has to respond to subsequently absorbed photons and detect changes in illumination. Low level of intracellular calcium ions due to cGMP-gated channels closure leads to activation of rhodopsin kinase, which phosphorylates metarhodopsin 2, reducing its binding affinity for transducin. Then arrestin completely deactivates the phosphorylated metarhodopsin 2, capping its residual activity, followed by several intracellular enzymatic processes leading to inactivation of transducin and cGMP phosphodiesterase. Low level of intracellular calcium ions also activates guanylyl cyclase activity, which synthesizes cyclic GMP. The change in configuration of opsin causes its absorption spectrum to shift into the ultraviolet so that the pigment loses its visible color. That is why this process is called bleaching. Bleaching limits the degree to which the rods are stimulated, decreasing their sensitivity to bright light and allowing cone cells to mediate vision in daylight conditions. The change in configuration of opsin also causes dissociation of retinal from it. The all-trans retinal, after dissociation from the opsin, is reduced to all-trans retinol and is transported into the pigment epithelium, where appropriate enzymes ultimately convert it into 11-cis retinal. This latter is transported back into the outer segment, where it recombines with opsin in the discs. This process is called the visual cycle, allowing the regeneration of rhodopsin. It takes place in darkness and is central to dark adaptation when rhodopsin levels depleted from bleaching in a bright environment increase, enabling rod cells to become sensitive to dim light again. All transretinal needed for normal functioning of rods cannot be synthesized by human pigment epithelium and must be supplied by vitamin A in the diet. The cycle using the pigment epithelium supplies chromophore to both rods and cones. However, this pathway is slow and cones use mainly a separate mechanism for recycling of chromophore. The all-trans retinal, after dissociation from the opsin, is reduced to all-trans retinol, as in rods, and is transported into Muller cells, where it is converted into 11-cis retinol, which is transported back into the cones. There, 11-cis retinol dehydrogenase converts it into 11-cis retinal. This pathway satisfies the greater chromophore demand of cones and provides their rapid and complete dark adaptation. A small population of non-canonical ganglion cells expressing the photopigment melanopsin was identified and named intrinsically photosensitive retinal ganglion cells. Melanopsin in varying quantities is contained along their cell membrane, including soma, axon, and dendrites. Several studies show that intrinsically photosensitive retinal ganglion cells serve non-image forming responses, including contrast sensitivity, pupillary light reflex, circadian photoentrainment, light aversion, sleep, and mood. Moreover, in the developing visual system, these cells influence cellular targeting, migration, and circuit maturation. Thank you for attention.